The light-dependent reaction takes place within the thylakoids. It begins with photophosphorylation, which utilizes energy derived from light to produce ATP, and as stated, is carried out in the thylakoid. The next step is photoactivation of photosystem 2. This is the first part to be activated by light, and it contains two special chlorophyll molecules that will absorb any energy from a photon of this light. The chlorophyll pass on their energy, and in turn, electrons within it become excited. Next, two electrons are donated that reduce plastoquinone, which is a hydrophobic electron carrier within the thylakoid. Each individual chlorophyll will lose 2 electrons to produce 2 of these reduced plastoquinones, and this continues all the way into photosystem 1. Some important carriers in the electron transport chain, or ETC, are ferredoxin, which is the primary electron acceptor, and plastocyanine, which carries the de-energized electrons that left photosystem 1. There's also NADP, which is the ultimate electron acceptor of the electrons released from photosystem 2 in the photophosphorylation process. This brings us to the next step, which is photosystem 1. It is quite similar to the photoactivation of the first photosystem. The primary electron acceptor is ferredoxin, and the electrons that left photosystem 1 are replaced by the de-energized electrons carried by that plastocyanine. After this photoactivation, NADPH is formed using reductase. 2 molecules of the reduced ferredoxin are utilized to reduce NADPH, and this is done by the enzyme NADP plus reductase. After this, photolysis takes place. The formula for photolysis is 2H2O, which turns into 4H plus 4E minus plus O2. Basically, it is the splitting of water molecules into oxygen, hydrogen ions, and electrons in the presence of light. Light causes the loss of electrons from the reaction center of photosystem 2, which then acts as an oxidizing agent to trigger the reaction. Oxygen is a byproduct of this photolysis reaction and is also the first step in cellular respiration. The hydrogen ions are then used for chemiosmosis, and the electrons are used in the light-dependent reaction, as it constantly replaces electrons lost by photosystem 2. The proton gradient is also important, and it is resulting from this higher concentration of protons, providing the potential energy for the ATP synthase complex to drive the production of ATP, just like in cellular respiration. Finally, chemiosmosis involves the pumping of protons, or hydrogen ions, into the intermembrane space of thylakoids by using energy released by electron transport along the ETC, followed by diffusion of protons into the stroma down a concentration gradient through ATP synthase to produce ATP.
The Calvin cycle takes place in the stroma. The first part of the Calvin cycle is carbon fixation, which creates all the carbohydrate compounds found in photosynthetic organisms. It starts by adding CO2 to ribulose biphosphate, which is then catalyzed by the enzyme ribulose 1,5-biphosphate carboxylase, also known as Rubisco for short. Each turn of the Calvin cycle requires 2 molecules of ATP and reduced NADPH. After 3 turns of the Calvin cycle, 1 molecule of triose phosphate is produced, which consequently can produce glucose and other carbohydrates. Reduction of glycerate 3-phosphate 3-PGA and 3 carbon atoms to triose phosphate 3 carbon atoms. Then glycerate 3-phosphate is created by the addition of carbon and oxygen. The 2-to-1 ratio of hydrogen to oxygen is reduced, and hydrogen has to be added, which involves both ATP and NADP. That was produced in the light-dependent reaction. ATP provides the energy needed to perform the reduction. NADP provides a hydrogen atom. The triose phosphate is used to regenerate RUBP and produce carbohydrates. 2 triose phosphates can undergo a series of reactions to produce glucose molecules in the form of starch and other organic molecules, and the majority of the triose phosphates has to be used to regenerate RUBP. For the Calvin cycle to continue indefinitely, RUBP must be produced as much as it is consumed. This requires ATP. One triose is used towards glucose or other organic compounds, and 5 triose are used to regenerate RUBP.